This is my new video regarding the test for the differentiation and identification of the aldehydes and ketones. And let's start the video. Test for the carbonyl group. Basically, this is a test in which we can detect the carbonyl group, and it means that this is a test for both aldehyde and ketones. So if we react the aldehyde or ketone with 2,4-dinitrophenylhydrazine, it is an example of condensation reaction. In this condensation reaction, actually two molecules join together, and you can see here this is an aldehyde and this is 2,4-dinitrophenylhydrazine. When they combine together, there is the formation of 2,4-dinitrophenylhydrazone. Its color you can see in the next picture is deep red color. So when 2,4-DNPH react with any of the aldehyde or ketone, it will give a deep red color like this. This is the formula, as you can see here, is for 2,4-dinitrophenylhydrazine. The precipitate formed here, this precipitate, can be recrystallized and its melting point can be measured experimentally. The identity of the precipitated compound can then be found by referring to melting point data. From this, we can identify the specific aldehyde or ketone used in the test. So now how we can distinguish between the aldehyde and ketones? There are actually two tests for them, which are the Tollern reagent test and the Fehling solution test. First of all, testing with the Tollern reagent. So Tollern reagent is aqueous solution of silver nitrate in excess ammonia. So it is also known as ammonical silver nitrate solution. The silver ions act as a mild oxidizing agent. When warmed with the silver ions, it oxidizes aldehyde to form a carboxylate ions. Under alkaline condition in the test, any carboxylic acid formed is immediately neutralized with a carboxylate ion COO, as H plus is removed from the COH and salt is formed. In the redox reaction with aldehyde, the Ag plus ions themselves are reduced to silver atoms. These silver atoms actually deposited on the walls of the test tube, as you can see here. So that's why we call it as Tollern reagent test or silver mirror test. There will be no change observed when a ketone is formed with a Tollern reagent, so no redox reaction takes place. It means this test can only be given by an aldehyde and not by the ketones. Now testing with the Fehling solution. The Fehling solution is an alkaline solution which contains copper ions. When warmed with aldehydes, the copper ions act as an oxidizing agent and are reduced to Cu plus ions. The clear blue Fehling solution turns to opaque red, like here you can see here. So as with the Tollern reagent, ketones are not oxidized in this test, so there is no change. The Fehling solution remains blue when warmed with ketones. It means that both Tollern and Fehling solution test can only be given by the aldehydes, not the ketones. Now reactions to form triiodomethane. This is a very important test and a lot of questions are used to come on the basis of this test. Triiodomethane or iodoform is form a yellow PPT with methyl ketones, that is compound containing CI3CO group. You can see the yellow PPT of iodoform or triiodomethane. Now note that ethanol CI3CHO and aldehyde also contains CI3CO group, so it will also give this test. Chemists use appearance of a yellow PPT as evidence of CI3CO group in unknown compound. The reagent used is an alkaline solution of iodine which is warmed together with the substance being tested. Now you can see in this equation a methyl ketone first convert with the help of alkali and iodine, convert into RCOCl3 and then into carboxylate ion along with the formation of triiodomethane. And how we can be sure that this is triiodomethane? When separated from the reaction mixture, the yellow crystal of triiodomethane can be positively identified by their melting point, which is 119 degrees centigrade. Reactions at triiodomethane, it means with respect to alcohol. Now see this one. This is a kind of alcohol which contains CH3CHOHR. So only alcohol with above mentioned structure can give triiodomethane test. So it can be given by secondary alcohol, hydroxy carbonyl compound, and which contain 3 halogen atoms in the CH3 methyl group replaced by iodine atoms. The intermediates hydrolyzed to form yellow precipitate of triiodomethane, CHI3, and sodium salt of carboxylic acid. Ethanol is the only primary alcohol give triiodomethane test. No tertiary alcohol can give triiodomethane test, but as I already mentioned, secondary alcohols can give triiodomethane test. Now we have different questions from different papers. Like in this one, what could be the answer? Basically, a yellow PPT of Ca3, Ci2, CO2 ions is formed. It is written here. It means that main ketone should have 4 carbon atoms, out of which 1 carbon atom used to make CHI3. Secondly, CHI3 is the correct formula, so answer is A. Second question. Everything is clear. Secondary alcohol give ketone with hot acidified manganese. This is one of the important thing, and you should know that hot acidified manganate is in fact oxidizing agent. Second important thing, the propanol alcohol use only 5 moles of the oxygen, while the butanol alcohol use 6.5 moles of oxygen. So obviously this is correct. So answer is B. Here it is written CO2. It is not CO2, it is actually O2. In this third question, compound 1 is an aldehyde, ethanol, which shows all tests with iodine, Fellings, and tolerance agent. Compound 2 is a ketone giving CH3CO group, so it can do iodine test but not other tests. Compound 3 is a ketone and do not contain CH3CO group, so cannot show any 3 tests. Answer is A. In this last question, you can see here in the question, the tolerance agent convert aldehyde to carboxylic acid. If you remember, I have just mentioned at the start of the video, NaBH4 is a strong reducing agent. It adds hydrogen with all oxygen atoms. As you can see here, hydrogen is added. Sodium dichromate is an oxidizing agent. So that's why this one, B part or A part, they are both incorrect. So answer is D. Thank you very much. This is the whole video, and I will be back with another video. Thank you. Allah Hafiz. See you in